let's look at the first step of how we've kind of touched upon this already, but we're going to see it again in the context here of recombinant DNA technology. How can we isolate a gene out of a chromosome in order to be able to work with that particular gene only and insert it into another organism, such as a bacterium or another type of organism? So, as we said, again, we use this technology of restriction endonucleases or restriction enzymes. So we said that, so for example, this here is our gene of interest that we want to isolate. We have to know, we have to investigate the DNA sequence before and after our gene in order to identify whether there are recognition sites for a particular restriction endonuclease out of the many that exist. I then actually mix my DNA with that particular restriction endonuclease here in orange, restriction enzyme, and the enzyme recognizes the DNA and cuts it at that particular location. So the restriction endonucleases, some of them, not all, have a unique ability to be able to cut these DNA sequences in quite a peculiar way. So let's say that this is the sequence that they recognize, and they will cut it here and here, thereby creating what we call sticky ends. What are sticky ends? They're a little short overhang, basically, of single-stranded DNA. And why do we call them sticky? Because they will have the tendency to anneal again with their complementary sequence. We can then add promoter regions or terminator regions. Why do we do that? Because before we actually go and insert this gene into the organism that we want to make, that we want to genetically modify, we want to also ensure that this gene is going to be expressed at the appropriate levels. So we may have to add a promoter region. Promoter should read. Other methods of isolation are also possible, such as using a PCR. Because again, remember that, again, we're going to have our gene of interest, and I could, but the PCR would only be suitable if my gene of interest is less than 1,000 base pairs long. So it's not always possible to use PCR. I can also use reverse transcriptase. What is reverse transcriptase? It is an enzyme which converts mRNA back to DNA. So I could actually, if I'm interested in the genes that a particular cell or tissue expresses, I could isolate mRNA from that cell or tissue and then use reverse transcriptase to synthesize it back to DNA. Okay, so once I have isolated my gene, I have to insert it into a plasmid. So we call the plasmid a vector. A vector is a piece of DNA, such as a plasmid. It can also sometimes be a virus that is basically going to transfer my gene of interest into the organism that I want to modify. In this particular case, we are looking at bacteria. We will cut the plasmid open, but we will crucially have to use the same restriction enzyme as we did in the gene of interest that we generated previously. Why is that? So here, for example, in blue is my gene of interest, and here, other way around, sorry, other way around. Here in blue is like a little zoom in. So I'm basically zooming in here, right? Of the region of my plasmid. My plasmid will also happen to have recognition sequences for the same restriction enzyme, because as we said, these recognition sequences are really common across DNA. So my enzyme generates these sticky ends, as we said, these single-stranded ends. And I will use the same restriction enzyme. So here we are zooming in. Here, I have done that previously to cut out my gene of interest from the chromosome that I took it from. So my gene of interest and my plasmid basically have the same sticky ends. What does that mean? That when I mix them together, The sticky end of my plasmid is going to anneal to the sticky ends of my genes, of my gene of interest, and they will become one continuous piece of DNA. This stage here is called annealing. And the annealing is achieved by an enzyme called DNA ligase. Some of you might recall that DNA ligase is an enzyme that was also involved in DNA sequencing, particularly of the strand that is synthesized in a non-continuous way in fragment. DNA ligase catalyzes the formation of a phosphodiester bond. So it catalyzes the formation of the sugar phosphate backbone, thereby making two pieces of DNA completely continuous with each other. Permanently incorporates one gene into the plasmid. Note that DNA polymerase also catalyzes the formation of a phosphodiester bond. The difference is that DNA polymerase adds one nucleotide at a time. DNA ligase can ligate two already long pieces of DNA and double-stranded piece of DNA. What is the next step after I have taken my gene of interest and I have inserted it into the plasmid? I want to then finally insert that into a organism, which I will call the genetically modified organism. In this case, a bacterium. And this process is known as transformation. So I have transformed, this is a transformed bacterium. Transformed bacterium. And it is also at the same time, of course, a genetically modified or transgenic bacterium. How will I do that? First of all, I need to create some kind of instability or permeability in the cell membrane of the bacterium, in the cell wall and the cell membrane. Cells are normally quite reluctant to be able to take in pieces of DNA from their environment. So I can do this permeabilization, as it were. This is what the dotted line is supposed to be showing here. So this membrane is permeable. And I can do this in a couple of different ways. And depending on the exam board here, you're going to have to quote different ways. So one way is electroporation, which you need for AQA and OCR, which means that you literally apply a little electrical current onto the cell, which is not going to kill it, but it's going to create pores in the membrane. Or you can add a solution which contains calcium ions. And calcium ions are also going to permeabilize, create pores in the membrane. And you will also create, because they're positively charged, there's going to be a little bit of repulsion between the plasmid DNA and the cell membrane. The same restriction, example section, sorry. Explain why the same restriction enzymes must be used during genetic modification when cutting a gene and a plasmid. When a gene and a plasmid, sorry, is to be ligated into. So my context here is the genetic modification. My command word is explain. However, it's only worth 2 marks, so it's going to be a short explanation. And my direction is that I am going to discuss the importance of how restriction enzymes cut DNA. Take a minute to okay. So the same restriction enzyme is used, if we use the same restriction enzyme, it means that both the gene of interest and the plasmid will produce identical, this is the key here, sticky ends. That is my 1 mark. The sticky ends are therefore complementary to each other, allowing DNA to anneal and allowing, here you could add a little bit of specificity. This answer is fine, it's according to the mark scheme. You could say allowing gene and plasmid, if you want, to anneal.